Hello, everyone. My name is Nusrat Tamzaman. I'm one of the founders of Sinoveda. We are a pharmaceutical company in Edmonton. And it's my absolute honor for me today to introduce Dr. John Lewis, who is an Edmonton celebrity. I think you will all agree with me. And we have been reading about him a lot lately in the media. And we all know why, and it is an absolute privilege for us to have him with us today, and we are going to hear from him directly. Dr. John Lewis is CEO of Entos Pharmaceuticals, an Edmonton-based biotechnology company developing genetic medicines using the Fusogenics drug delivery system. Entos is developing DNA-based vaccine and therapeutic countermeasures for COVID-19 using the Fusogenics platform technology. Dr. Lewis holds the Frank and Carla Sojonki Chair in Prostate Cancer Research at the University of Alberta and is a professor in the Department of Oncology, where he chairs the Alberta Prostate Cancer Research Initiative. Dr. John Lewis's research interests lie in the areas of nanotechnology and imaging as they relate to infectious disease as well as chronic diseases such as aging and cancer. His group is focused mainly on the study of the spread or metastasis of cancer using advanced live imaging techniques. So without further ado, I'd like to please welcome Dr. Lewis to share his work with us today. John, over to you.
Well, that was just a wonderful introduction, Nusrat. Really appreciate it. And thanks so much for the organizers of this fantastic conference for inviting me to speak. I'm going to share my slides. Yeah, so it's a great pleasure to be here today to share really just a Herculean effort by my team over the past year to try to, leverage some of the technologies we've developed at Entos Pharmaceuticals to get us out of this pandemic. Entos Pharmaceuticals, as Nusrat mentioned, is an Edmonton-based company. I'm a professor at the university and run a lab there. For many years, working with the team and collaborators, we've been keenly interested in bringing to bear novel genetic technologies to create cures for diseases like cancer, and now COVID-19. I want to share today a little bit about the platform we've developed, our vision for it, and our work specifically on vaccines for COVID-19. Like I said, Entos is an Edmonton-based company. We are so we work out of the Cates building at the University of Alberta. We've got a team there. We're also in the Merck Invention Accelerator at Tech Labs in downtown Edmonton. We also work very closely with the Alberta Cell Therapy Manufacturing Facility at the University of Alberta, where I'm happy to say we just completed our first GMP manufacturing run, which is clinical grade manufacturing run of our very first in-human product, this COVIDgenics vaccine. And we've got a dedicated team of scientists at all levels, PhD grad students and senior scientists, have been working really day and night in this pandemic to bring this into clinical trials. We're also in the Johnson & Johnson Innovation Labs in San Diego and have labs and offices in San Diego. So, what makes a safe and effective vaccine? Well, I think we've all heard a lot about what that might be. In our minds, we wanted to develop something that activates and engages multiple arms of the immune system. And you probably heard a lot about antibodies. You probably heard maybe a little less about T cells. The key here is that we believe both are very, very important. So innate immunity is sort of the, initiates and shapes the adaptive response that comes a little bit later. This has been shown to be key in the protection, particularly of children against COVID-19. But really, vaccines seek to stimulate humoral immunity, which is an antibody-based response, that takes a few weeks to develop, but then your body is able to generate very specific antibodies that recognize the pathogen and clear it before it can create a significant infection. On the other side of things, we have cellular immunity. So we produce T cells of different types that are able to seek out find virus-infected cells in your body and effectively recognize them and clear them. And the dynamics and the effectiveness of both of these arms of the immune system have slightly different properties, and it's really critical to have both of them engaged with a vaccine to provide initially protection against the initial insult, and then long-term durability of protection over time. And so what you probably learned through this pandemic is that the genetic vaccines have really emerged as really key tools in the fight against COVID-19. The RNA vaccines now approved by Moderna and Pfizer are examples of genetic vaccines. Genetic vaccines basically deliver genetic instructions to our own cells to be able to produce the vaccine in our own bodies and display that and create the immune response. And so the idea here is that we have a vaccine that delivers the instructions to create the coronavirus proteins, and then your body produces the immune response against it. What's important about genetic vaccines is that because now your own cells are making the vaccine, this is made inside your cells. And so it's processed in a way, the proteins are processed in a way that delivers them both to create an antibody response and a durable T cell response. And with genetic vaccines, we can also do some very creative things with adjuvants, or things that improve or shape the immune response. And I'll talk a little bit about that as well. So I'm going to make a provocative statement now, particularly after Ken told you yesterday about Providence and the RNA vaccine. We think DNA is the ideal payload for genetic vaccine, and I say that with a smile on my face because clearly RNA is a winner in the fight against COVID-19. But we decided early on to go with DNA. We realized that RNA was going to be an effective countermeasure early on. We decided with our technology was uniquely situated to deliver a DNA vaccine, and we did this for several reasons. So like RNA, DNA carries the blueprints that makes these antigens inside cells and creates a strong antibody and T cell response. But unlike RNA, DNA is much more thermostable. So we knew with DNA we could create a formulation that could be stable in the fridge for long periods of time, more than a year, and even at room temperature for long periods of time. The other thing is the way DNA makes antigens is a little bit longer lasting than RNA. RNA is very potent and very short term, and so we thought DNA might be a good platform to create a single dose vaccine. And so if you look at sort of the different platforms that are available to deliver genetic medicines, they're mainly viral vectors and non-viral vectors like lipid nanoparticles. So viral vectors are like the adenovirus that the Johnson & Johnson vaccines or AstraZeneca vaccines are based on. And these do a good job of delivering genetic material. The issue is because they're a virus, they elicit their own immune response. And so your body also learns to recognize the virus itself, makes it very, very difficult to deliver more than one dose effectively. On the other hand, we have lipid nanoparticles, which are a great platform for encapsulating RNA, and we've shown this astounding efficacy with the early COVID-19 vaccines. And the issue here really is the fact that they don't do a great job of delivering DNA. They actually become quite toxic, and so we sought to develop a platform that allowed for the delivery of DNA. This is, I'm a professor, so I have to show you a slide teaching you all about LNPs. But I think what I want to communicate with this is that the key ingredient of lipid nanoparticles that allows for the delivery of RNA are these positively charged lipids. So LNPs are basically groups of lipids encapsulating in genetic material. The positively charged lipids actually do the work of delivery, and but these are sort of bring the weakness for DNA delivery. So we thought the ideal LNP for DNA delivery would be one that uses an alternative mechanism of action. And so working with Entos co-founder and Canadian scientist Roy Duncan, he's a professor at Dalhousie University. About 15 years ago, he discovered this class of orthoreovirus, a fusogenic orthoreovirus. It's a mouthful, but really does a really remarkable thing when it infects cells. He found 15 years ago that when it infects cells, it causes all the cells around it to fuse together. And he immediately realized the significance of this, that it produced a novel fusion protein. What he didn't realize at the time is how unique it was. So I'm showing it here beside the influenza HA fusion protein. This is a huge multi-subunit immunogenic, which means it creates its own immune response protein. It does fusion, but these fusion-associated small transmembrane proteins that Roy discovered are tiny, 100 times tinier. single transmembrane domain, tiny ectodomain, non-immunogenic, importantly though, sufficient to induce fusion, and particularly efficient at producing or inducing fusion when you incorporate them into a nanoparticle like a vaccine. And so Roy and my labs have been spending the last decade doing structure function analyses of related fusion proteins from different viruses. And now we've got a fully synthetic hybrid version of a fusion protein that we incorporate into the platform. It's really, I would call it LNP version 2.0. We call it a proteolipid vehicle. And it's really an LNP minus those positively charged lipids, but now including this fusion protein. And we've worked a lot with our partners, Precision Nanosystems in Vancouver, for instance, at developing an optimized single-step microfluidic manufacturing process under GMP in the Alberta Cell Therapy Manufacturing Facility, where we can ostensibly scale up very quickly to millions of doses of vaccine. And so we've optimized this platform for the delivery of RNA, for the delivery of gene editing tools, and importantly for this project, the delivery of DNA. And again, we're using this scalable microfluidic platform. It's really amenable to, starting on the benchtop in the lab on a scale of, maybe a few drops, and using the same microfluidic chip, we can go to complete automated GMP manufacturing, continuous flow, and do, 2,500 liter batches of drug product. And we have this GMP manufacturing up and going in Edmonton, and we're looking forward to scaling this up as we move this vaccine to the commercial market. So when we designed this vaccine, we really had several considerations, and this is the main reason for selecting DNA. Stability is key, and we've seen from the rollout of the vaccines already, requiring deep cold, it's been really challenging to really administer them outside urban areas. So we wanted stability in fridge temperatures for a year, room temperature for a month. We thought the plasmid DNA would allow us for single dosing. We thought durable T cell response was really key. We know from individuals who recover from SARS-CoV-1 that they still have T cells floating around that recognize the disease and are protective. We have two candidates. One is a pan-coronavirus candidate that also, we have these variants that are emerging. And so this candidate aims to go after all emerging variants and potentially other coronaviruses. And again, we have this scalable manufacturing. So we spent the early part of last year going through 24 different candidates, and we decided to advance these two candidates last June to clinical development. So VAX-001 is very similar in concept to the Pfizer and Moderna vaccines. It encodes the full-length coronavirus spike protein. We've inserted it into what's called a nanoplasmid. It's a really advanced, minimal DNA construct that has a lot of clinical experience and is safe and effective in other products. And we use these creative genetically encoded adjuvants, double-stranded RNA, CpG, and RIG-I to really potentiate that antibody and cell-based response. And we really selected this one, obviously, due to the success of the RNA vaccines and our strong preclinical data. We also have our second candidate is a little more sophisticated. It contains actually machine-learned epitopes, 34 of them, from not only the spike protein, but also the E and M proteins from coronavirus. These are highly conserved proteins, over 96% between the different closely related coronavirus types. And so we expected this vaccine to be effective not only against the variants, but also against other coronaviruses. So this is thinking forward to the future and future pandemics, again, encoded in a nanoplasmid with the adjuvants. And so we spent the better part of the last 6 to 8 months doing the preclinical studies, looking in multiple animal models of the effectiveness of these vaccine candidates. And so the data I'm going to first show you is looking at just the antibody production, not looking at the how effective the antibody response is. But so for VAX-001, with a 2-dose regimen spaced 14 days apart, and at 3 different dose levels, 25 micrograms, 100 micrograms, and 250 micrograms, we see 100% seroconversion, which means 100% of the animals had at least a 4-fold increase in spike-specific antibodies. And what was interesting, you compare it to other DNA vaccines like Inovio's vaccine, where they're giving 2 milligrams per dose. We're giving just a fraction of that dose and getting a similar response. VAX-002 is this multi-epitope pan-coronavirus candidate. Again, every animal we dosed at every dose level had seroconversion. And when we looked at the B cells in those animals, we saw that we're getting a potent stimulation of the B cell compartment, which indicates we're engaging that antibody production system. So we know we produce antibodies, we have seroconversion. Are these antibodies neutralizing? Do they effectively prevent coronavirus infection? So in this pseudovirus virion assay, again, an experiment where we gave 2 intramuscular doses 2 weeks apart, looking at a very sensitive assay with luciferase, and comparing to a panel of convalescent patients, so patients who had been infected with coronavirus in some case and had recovered, and so have sustained antibody levels. We found that at all doses, we got as good or better neutralizing titers of these antibodies compared to convalescent human sera. And what's really important is we've also been looking at some of these variants. So the 614G variant is the one that's really spread all over the world. And it's in pretty much every variant, including the UK and the South African variant. We found that actually the convalescent human sera is much less potent at neutralizing this particular variant, and that both the VAX-001 and the VAX-002 did a good job of neutralizing these variants. And we're currently looking at the other emerging variants that are coming, and those the number of those variants is mounting pretty much by the day. So this is, the first part of the most exciting data I want to show you. So this is neutralizing antibody response, but now we've done it in non-human primates. So in this experiment we looked at basically comparing at multiple dose levels, either a single dose or 2 doses of the Covagenics VAX-001 vaccine, and then looking at neutralizing titers in our neutralization assay. And so what we found was actually pretty remarkable. Whether we gave a single dose at day 0 or 2 doses spaced 1 month apart, the onset of the neutralizing antibodies against SARS-CoV-2 are basically lockstep. So we're seeing equivalent neutralizing titers. And I show the red line here. This is the expected level required for protection from infection. We're well over that level and increasing over 42 days. So we're a lot of optimism here that our vaccine could be used with a single dose. So the one thing I mentioned I think is really important is for durability especially. So the neutralizing antibodies may wane over time, but again, we think that stimulating a potent T cell response is critical. So VAX-001 induces a significant and potent immune response that's antigen specific to the spike protein. And then when we look at the sort of the nature of that immune response, we find, and I'm just going to show a subset of the data here, we find that CD8 positive cells are specifically stimulated and proliferate in response to antigen. So we're getting a CD8 response. Those CD8 cells, are more active, express more interferon gamma. And most importantly, there's a significant increase in that compartment that expresses CD69, which means these are active, engaged killer T cells. We've actually taken this farther, and what I think may be an industry first is that we've now looked at the functional impact of these CD8 positive killer T cells. So we've created a functional CD8 killing assay where we take T cells from immunized animals and incubate them with target cells that express the SARS-CoV-2 spike protein. And we see a dose-dependent and significant increase in the ability of those T cells from immunized animals to kill those target cells. So really, really encouraging there. Okay, so that brings us to today. I've showed you all the preclinical data. We've been working really hard. So imagine this vaccine that we're producing is an LNP, but it's also a protein-based vaccine. So we've really had parallel programs to create GMP protein and GMP LNPs and combine them together to create a fusogenics vaccine. I'm happy to say that we completed our very first batch of final GMP vaccine for clinical trials. We're poised and ready. That vaccine is actually on its way to Halifax to start the first in-human phase 1 clinical trial. Now, thinking about the speed at which we need to move to get an approved vaccine through, we've got an adaptive phase 1, phase 2 clinical trial design. So this means that we're going to be recruiting multiple cohorts of patients, including 36 young healthy adults and 36 elderly, but that the completion of the phase 1 portion isn't gated on that entire population. So as soon as we get safety data from that first 36 patients, we'll be asking Health Canada to allow us to move to the phase 2 portion. And in the phase 2 portion, we'll be looking at 2 different dose levels. But most importantly, we'll be also looking at the impact on immunogenicity of either 1 dose or 2 doses before we move to the phase 3. Now, obviously, we're in a dynamic environment where multiple vaccines are becoming available, and so we're hoping that by the time we're at the phase 3, correlates of protection or what could be levels of neutralizing antibodies or levels of T cell response that indicate effective protection will be established, and that'll allow us to move ideally seamlessly into the phase 3 portion before the end of this year. And so we completed a lot. We're less than a year into this program, and we've completed, the team has been working just night and day. So we selected our lead candidates June of last year. We've been successful in getting some funding. I've estimated that it probably costs $350 to $500 million to take a vaccine from beginning to end. So we haven't received that yet, but we're happy. We got $4.2 million from CIHR, $5 million from NRC IRAP through federal funding. We're looking forward ideally for IRAP to support the next phases of the clinical development. We've completed what's called IND enabling or CT enabling toxicology in rats that was worked out well. That was completed in December. Challenge studies, really huge demand around the world for these studies. These are ongoing. We should have those results shortly. We did complete our first GMP batch of the vaccine that's heading toward the clinical trial site, and we're looking forward any day to starting that adaptive phase 1, phase 2 trial in Halifax, just waiting for Health Canada go ahead. And I just want to end the talk by saying, we've been living with this for a year, but I think, it's been talked about that it's a race to a vaccine, but I think it's really a marathon. The first wave of vaccines have, it's wonderful that some of them are very effective, but as we see these variants emerge, I think we're going to see differential protection against variants. We need to, make up to 16 billion doses worldwide, not even including boosters. So I think we're going to be in this for a while, and I think we've been really pushing. I think we need a made in Canada solution for this because we're seeing the rollout to Canadians, is frustratingly slow. And I think if we invest, there are plenty of great vaccine candidates in Canada, Medicago, Providence, PNI, others, even locally with Michael Houghton's vaccine. I think, an upfront and substantial investment in Canadian innovation could really build this industry in Canada and provide security for Canadians as we work our way through this pandemic. And I think I'm really gratified to see, for this industry that we're witnessing the dawn of genetic medicines, and Entos and the team feel really privileged to be a part potentially of making an impact. Thank you so much for your attention.
Thank you, John, for the wonderful presentation. And we are really proud of the work that you are doing. And is there any questions? I don't see any here. Rob? I don't see any questions, but I guess I have one for you, John.
Sure.
Most companies are doing the mRNA technique, and you're doing the DNA, and looking at some of the advantages that you talked about with regards to the stability and also the dosage regimen. If you can go with just 1 dose, that would be fantastic. But do you foresee any other problems in the clinical trial? And if so, what would that be?
Yeah, no, so I think that, so DNA vaccines have really been around since the early 90s when they were first shown in animal models to be effective against infection of flu. So I think the promise of DNA vaccines have been around for a while. I think we've seen DNA vaccines move forward by companies like Inovio and throughout the world. The challenge is they require huge doses. So 2 milligrams per dose, 3 doses. Inovio requires an electroporation, electric shock device. Others require sort of a propelled injection technique. So, I mean, we're really bringing LNP technology to the delivery of DNA, dramatically reducing the doses. So I think, if we find in clinical trials that it works as well as it does in the preclinical, I think it'll be a great alternative to the RNA vaccines.
Okay, thank you. I think we have one question here from Sami Salama. He wants to congratulate you and your team for the incredible research you are doing. And Entos is setting an example for all the university-based researchers on how to landscape from lab to fab. What is your advice to other university-based researchers?
Yeah, I mean, it's a big question. I think there's definitely so obviously university-based researchers should focus on what they do best and not try to do things they don't do well. I think for many academic researchers, creative-based discovery is the key to innovation in Canada and forwarding our academic goals. That said, though, for university researchers that are interested in translating findings to the clinic, I mean, I think it's just a matter of getting out there. We have fantastic support structures and groups within Edmonton and throughout Alberta to learn about this. We're seeing a huge increase in accelerators like the Merck Accelerator and JLabs and CDL, for instance. My advice would be to get out there and network and talk to people who've done it, and we're all here to help.
Okay, thank you. Another question from Bryce McNaughton. Assuming phase 1-2 trials proceed successfully and as quickly as possible, how will you manage phase 3 trials when most of the Canadian public is already vaccinated?
Yeah, Bryce, that's an excellent question, and one we've been sort of thinking about, since the first vaccines got emergency approval. So certainly we think, based on the speed of vaccination of Canadians, that we'll be able to get to the end of the phase 2 portion. That said, though, even in phase 2, we're working with a CRO that has sites in South America, India, Australia, and other places. So we may even have to open phase 2 sites outside of Canada. Phase 3, we're looking at there's a couple ways we can do it. So we can do a comparative study where participants would get an approved vaccine or our vaccine instead of a placebo control, and then we could look at doing that phase 3 outside of Canada, which could be a possibility. And I'm hoping that Canadians will be vaccinated by the time we get there.
Okay, thank you. I think we are almost out of time, so just one question from me, just a general one. If there was just one piece of advice you could give today to all the entrepreneurs in the room that are in their journey and not quite there yet to achieve their goals, what would that be?
So I would say there's nothing wrong with taking critical advice, but in the end, you have to just forge forward as fast as you can. You don't get to where you're going without working to get there.
Okay. Thank you. I think with that, on behalf of BioAlberta, I would like to thank you, John, for taking time and sharing your work with us. It was just incredible, and wish you all the best and success with your vaccine launch. Thank you. Over to you, Rob.